First off, I would like to acknowledge the work of my collaborators, Jack Zinc here at the University of Alberta, and several other collaborators who have helped me complete this work. Stuart Hameroff at the University of Arizona, Rudy Tanzi at Harvard, Lee Goldstein at Boston University, as well as Noel Casey, and Deepak Chopra of the Chopra Foundation. I'd also like to acknowledge the funding agencies that have provided the money to get this done, several from both the US and Canada. And finally, I would like to thank the conference organizers of Toward the Science of Consciousness 2013 and the Dalibag Educational Institute for inviting me. Thank you very much. First off, what is memory? Basically, it's defined as the ability to encode, store, and then retrieve information, retrieving being one of the most important points. In our brains, this is postulated to be the result of synaptic connections. So our brain is made up of billions of neurons, and the connections between these neurons, the strength of these connections, the number of these connections, is attributed to memory formation. This is known as Hebbian theory. It's also called synaptic plasticity. So the changing of the strength of these synaptic connections is what gives rise to the formation of new memories. The main paradigm which describes this is long-term potentiation, the idea that neurons that fire together wire together, and those that don't fire together don't wire together. So in acting in unison, you can increase a connection between neurons. In firing at different times, you can weaken connections. However, while this is generally accepted as the standard paradigm for understanding memory, we don't really understand how this takes place on the molecular level. So we're talking about synapses between cells. How are these synapses strengthened? How are these connections strengthened? What is the molecular mechanism that underlies this? And most importantly, what is the substrate? What is the material that gives rise to this change in synaptic strength? We believe it's the microtubule cytoskeleton. We've heard lots of talk about microtubules. Now, here's a standard archetypal neuron. So you have your cell body with dendrites around the end. Signals come in from other neurons into the dendrites. They get integrated here, causing a reaction within the cell. An action potential propagates down the length of the axon and then out to connect to other neurons via their dendrites. Now, inside each one of these neurons, there's an elaborate architecture of microtubules. And this is an image that I stole from Stuart. I believe many people have stolen this image over the years. So you can see here that within dendrites and along the axons, you can have parallel alignment of microtubules or anti-parallel alignment in dendrites. And these are connected by other proteins called microtubule-associated proteins. And it holds these networks together and forms a structure within the neuron. Now, learning and memory involves a reordering of this microtubule cytoskeleton. So a shift in the connections between maps, between the lengths of microtubules and so forth. Now, many cognitive diseases that show memory impairment, including Alzheimer's disease, dementia, bipolar disorder, schizophrenia, among others that you heard Jack speak about earlier, these all show a dysfunction in the neural microtubule cytoskeleton. Synaptic plasticity, that changing of the strength between these connections between neurons, all involves changes in microtubule cytoskeleton. So in order to remodel, in order to change the strength between synapses, you need to remodel the cytoskeleton. So you can, like I said, change the cross bridges. These microtubule-associated proteins are maps between microtubules. As well, when you change these maps, you change the orientation of the microtubules, the way that they form together. This will also change the way that motor proteins in the cell are directed. So these tracks, these are microtubules within a neuron here. Along these, there's the proteins called motor proteins such as kinesin and dynein, which walk along the length of these microtubules, and they can go from the cell body out towards the axon or back and forth in the dendrites. And these things carry synaptic cargo, such as ion channels or vesicles filled with neurotransmitters. So by changing the structure of the microtubule cytoskeleton, you're changing the highway which these motor proteins travel down. And you can redirect the cargo within the cell to one synapse or to another. It's also been shown by the work of Nancy Wolfe that changes in this microtubule-associated protein arrangement, that these correlate with learning. So in order to get learning, there has to be a change. There's a degradation initially of these connections, and then a rebuilding of these connections during memory formation. As well, one other thing, dysfunction which affects learning and memory. So dysfunction of the cytoskeleton, like I said, in Alzheimer's disease can affect learning and memory, and many microtubule disrupting agents can affect memory as well. So anesthetics is one example. Anesthetics, here we're all familiar with them knocking out consciousness, but they also cause amnesic effects, so they can erase memory. As well, other microtubule disrupting agents can affect your memory. So here's a microtubule. We've seen this before. This is a microtubule, or a tubulin protein which makes up a microtubule. It's made up of an alpha monomer and a beta monomer. And off the ends of these, there's these tails that extend out, and these are called the C-terminal tails, which you heard Jack talk about earlier. And they're very important, so I'll be coming back to these eventually. Dimers form end-to-end to form long strands of tubulin protein. These are called protofilaments. And these protofilaments connect side by side and wrap around to form your microtubule structure. They're about 25 nanometers in diameter, about a micrometer to a millimeter in length. They're the most abundant structure, the cytoskeletal structure in neurons. And like I said, they're responsible for cell cargo transport, for maintaining the shape and orientation of the neuron, as well as influencing synaptic plasticity. Now, these things, again, because these are polymers, these tubulin dimers, they form up to form up a microtubule, but they go through a cycle of rescue and catastrophe. So GDP tubulin can attach to the end of a microtubule, making it longer. And then, for certain reasons, these things may rapidly fall apart and start to depolymerize, where all the tubulin falls off. And then that tubulin that is free can then exchange its GDP molecule for a GDP molecule, and then again reattach. So you get these cycles of growth and shrinkage in normal cells. In neurons, this cycle of growth and shrinkage is stopped by microtubule-associated proteins. So these things strengthen the microtubules and keep them fairly stable. This polymerization process is affected by many things, such as temperature, the pH of the cell, as well as ion concentration. And one of importance is zinc. So zinc in low concentrations can promote the assembly. In very high concentrations, it can cause aberrant formation, so causing microtubules to form in sheets or strange ring structures that are not conducive to normal neural function. So in Alzheimer's disease, the reason that we get this falling apart of microtubules, or what was originally believed, is normally in your neuron, the microtubule is stabilized by tau protein. Now, through some process, this tau protein becomes separated from the microtubule, and because the microtubule is no longer stabilized by the tau protein, it can fall apart. Or another interpretation is if a microtubule falls apart, then you're left with a bunch of tau protein floating around. And this tau protein then aggregates into neurofibrillary tangles. Now, in Alzheimer's disease, there's two major hallmarks. These neurofibrillary tangles, which are inside the cell, and then outside the cell, between neurons, you get what's called a beta amyloid plaque. Okay? Now, these are the two standard things that you see in every Alzheimer's brain. They're commonly accepted. They're signs of Alzheimer's disease, but there's no linkage between the two. They don't know how they relate to one another. Okay, so the beta amyloid plaques outside the cells, they correlate with cell death. So the more plaques you have, the more cells you have dying. However, the cognitive impairment, the loss of memory, correlates more with the number of neurofibrillary tangles seen within inside the cell. Now, because this link is unknown, there's no mechanism. Well, there was no known mechanism until I got working with Jack Stewart and the people down at Harvard and Boston. Lee Goldstein did some measurements of zinc within the brain. So here on the left, we have a regular mouse brain. On the right, we have an Alzheimer's model mouse. So it exhibits the same signs of Alzheimer's in a human. And the red color, the temperature scale that you see, is the concentration of zinc within the brain. So in a normal brain, you can see that it's highly localized here. This is the hippocampus. Right? And in the Alzheimer's brain, you can see that it's vastly spread out. So you get it spreading out in the hippocampus and then across the cortex. There's this normal zinc dyshomeostasis within the brain. Now, what Rudy Tanzi at Harvard had pointed out is that beta amyloid plaques strongly suck up zinc. So they take zinc when it's exuded from the cell, and they hold on to it. And they hang on to it, and they won't let it go. So what these plaques drive is this zinc homeostasis throughout the brain. Now, how does that relate to microtubules? Like I said, microtubules, the zinc can affect the way that they polymerize. So we formed models of tubulin based on the crystal structures of tubulin, and we predicted where zinc would bind. In low stringency modeling, we found up to 58 sites predicted with a false positive rate of about 5%. And this corresponds with some experimental evidence back in the early 80s. If we look at this in a more robust detail, we can find 6 high affinity sites where zinc prefers to bind. And of these, a couple are in the colchicine binding region, which you heard Jack speak of. Some are in the GDP binding site on the beta dimer. And another one is at a site which is known to promote the formation of these zinc-induced sheets. If we look at the electrostatics of these binding sites, here on the top we have a normal tubulin dimer with no zinc. Here on the bottom we have these zinc added at these sites. We can see changes in the electrostatic map. Okay, so red is negative, blue is positive. And normally these, if you look at these, the left side of the slide would sandwich together with the right-hand side. So this positive region here would line up with this negative region here. Now, if you increase the amount of positive in this region, you're going to increase the attraction between the two dimers, okay, in these regions down here. However, if you increase the positive regions up here, these would prefer not to join up together in normal microtubule arrangement. They would prefer to flip over to the sheet formation, which is consistent with what we've seen. So the idea is that at low zinc concentration, it would promote dimer assembly into microtubules. At very high concentrations, these things would prevent formation in microtubule arrangement, but would promote formation in this sort of zinc sheet arrangement. So this is consistent with what is known from experiment of microtubule polymerization with zinc. We built a kinetic model with a bunch of mathematical equations, rate equations, ordinary differential equations to describe how beta amyloid sucks up zinc and how that would affect the polymerization of microtubules. And these are our main results. So red is polymerized tubulin. Blue is the number of beta amyloid proteins that form together to form these beta amyloid plaques. And yellow is the free zinc that's available to microtubules for polymerization. On the left-hand side here is a percentage scale, so the percent of the value, an initial starting value. So initially you have only free beta amyloid, and you have all of your tubulin polymerized into microtubules, and you have a full amount of free zinc. As beta amyloid increases, so that as the plaques form together, you're sucking up more free zinc, and the number of microtubules within the cell will decrease to a steady rate. So what this is saying is that beta amyloid plaques induces zinc loss. This zinc loss drives the loss of microtubules. And this is completely independent of the loss of the microtubule-associated protein tau. So you don't need to have the hyperphosphorylated tau to lower the number of microtubules within the brain. So here, this is linking beta amyloid with loss of microtubules. Now, if you have less microtubules in the neuron, and you're still producing the same amount of microtubule-associated protein tau, what is this tau going to do? It's floating around free within the cell, and we believe that the hyperphosphorylation mechanism is a way to get rid of the tau. So then it forms up in these clumps, hopefully to get removed, but in pathological cases, it sticks around. So the loss of microtubules could drive neurofibrillary tangles. This would correlate with memory loss, an MT-based memory impairment. So loss of the memories correlates with loss of microtubules. And this would also link amyloid plaque pathology with neurofibrillary tangle pathology, which to this point has not yet been done. Now, microtubules, this kind of says that microtubules are very important to memory. Okay, so they're a good substrate. It lines up with some of these experiments. But what is the mechanism? How can you store information in a microtubule? This is just a protein polymer. So we did further work. This was actually preceded this work, but it led up to this. Whenever there's a neural event, so if you have an incoming action potential into a neuron, it opens up ion channels, which allows the influx of ions. And then this triggers events within the cell. So it activates certain proteins, it activates certain enzymes, and these enzymes go and they perform functions in the cell. One important enzyme is called calcium calmodulin kinase 2, which you heard Jack allude to, and it's vital for memory, and it's vital for long-term potentiation. A single point mutation, changing one amino acid within this enzyme, can result in memory impairment. Okay? And it's been suggested that this molecule itself is a molecular switch for memory. So by turning it on, you can store memories, and by turning it off, you can not store memories or even erase memories. And effectively, this records synaptic activity. So as there's an incoming signal, calcium is released into the cell, and this calcium wave is encoded in this enzyme. So normally the enzyme is floating around, and it's in this inactive state. It can't do anything. Calcium comes into the cell, binds with a protein called calmodulin, and this calcium calmodulin complex activates the enzyme, and it causes these regions, these green and yellow regions, which are called kinase regions, to be released. And these are now active. They can go and they can perform functions on proteins within the cell. And what they do is they phosphorylate the protein, phosphorylate a substrate, and they leave a tag on the protein. Over here on the far right, this is the electrostatic map for this protein. And you can see that it's fairly blue, which means it's fairly positive. Now, if we look at microtubules or tubulin are known to be phosphorylated by this enzyme. It's not known how, the mechanism is not known. But if you look at a single tubulin, it forms up into a microtubule. If you look at a patch of the microtubule surface, either in a B lattice here on the left or an A lattice here on the right, and look at the electrostatic potential of these patches, you can see that it's fairly negative. So we have a fairly positive enzyme, a fairly negative protein substrate. So you would suggest that these would have some sort of attraction together. Now, if you look at the size of a microtubule patch and this calcium calmodulin kinase enzyme, you can see that they're almost the exact same. So here's the enzyme, here's the patches, and here's an overlay of the A lattice. And you can see that it lines up almost exactly with this microtubule A lattice. In a B lattice, you would have to have a slight bit of flexibility in this enzyme, which we believe is perfectly available. I mean, you saw the legs extending out. They're held out by a string of amino acids, which would be free to move about with a little bit of strength. So depending on the way that these things line up, you can align these kinases with the surface of the microtubule in different patterns. Now, not every one of these kinases needs to be active. You could have one being active, you could have all 6 being active, or any sort of pattern. And it doesn't matter, I mean, you could have any sort of orientation. So this one could be active and this one, or this one and this one, or this one and this one. And you get all sorts of different orientations of the ways that these could align. Looking at the attraction between these two, between the electrostatic maps, we show a convergent field lines. The gray are the electrostatic fields between these two proteins. And they're converging, so they come together, which means there is a general attraction. If you look at the strength of this, it's approximately 6 kilocalories per mole at biological temperature, which is for a single kinase tubulin interaction. So you can have up to 6 times this amount for all 6 of the kinase. And this is a considerable binding energy, so it's fairly strong attraction between the two. Now, these kinases, like I said, can phosphorylate the tubulin dimers, and effectively this phosphorylation would then encode information. So because I said that there was different patterns, we can go through and form different patterns on the microtubule surface, and depending on which dimer which monomer is being phosphorylated, you can have multiple patterns forming, whether it's on an A lattice or a B lattice, effectively encoding information into the microtubule. Now, each one of these events between a single enzyme and the microtubule surface can encode somewhere between 64 bits and 5,218 bits. So each one of these events could be a fair amount of information being stored. And we're talking when there's a synaptic event, we could have many of these calmodulin microtubule, CAMK2 microtubule interactions. And each one of these events releases about 20 times thermal energy. Okay, so the binding, the attachment of this phosphorylation to the microtubule surface is very strong. So it's not something that's going to be attached and then the vibration due to the environment is going to cause this thing to be released. This thing is permanent almost. Now, what could this do? How can these things be read out? So this is the encoding mechanism that encodes it It stores it because it's fairly stable, but how do we read it out? Well, these phosphorylation patterns may tell, these post-translational modification patterns may tell microtubule-associated proteins where to bind. So it may tell where structural maps, which link two microtubules together, it may tell it where to bind, or it may tell where motor proteins where to walk. So here we have a kinesin molecule, and it steps down and will only step on a phosphorylated patch. And then when it reaches a microtubule-associated protein, it releases, and then releases from the surface of the microtubule. So this could effectively tell motor protein transport or tell the microtubule cytoskeleton how to form. Okay, so now we have we're reading out the information in the structure of the microtubule cytoskeleton or in the transport of these microtubule-associated proteins. Another way that this could be read out is via ionic propagation along microtubules. So microtubules, they can form a cloud of ions around them, and in the presence of potential differences, these form signals down the length of the microtubule. So this can also happen along maps. So in this particular orientation, if you have if this is one input, this is another input, and this center protofilament is our output read, then if you only have one input, you're not going to read anything on this center line. If you have one coming from the other, you have nothing reading on the center line. But if you have both, then the horizontal components can cancel, and you can get a signal traveling only down the length of the protofilament. So this is effectively an AND gate. Likewise, you can have something forming an exclusive OR gate. Right? So if you have it on one, it goes down the center. If you have it on the other, it goes down the center. But if you have both, and these are at a phase, and they can cancel one another. So this is an exclusive OR gate. And together, both an AND gate and an exclusive OR gate forms the basis for a universal logic. So any sort of binary logic that you have in a computer, you could form with these two gates on microtubule surface. So this effectively it forms a basis for memory. So you can encode it in microtubules, and then you can read it out. And this would have implications for neural function. Now, what about consciousness? Okay. This is much harder to define. If it was easy to define, we wouldn't be having these conferences. Now, we all accept that this is related to brain function in some way. And there's been arguments to relate consciousness and memory. There's got to be some mechanism linking memory and consciousness. If you're not conscious of an event, can you have a memory of it? Now, the most comprehensive extended theory of consciousness has been the Penrose-Hameroff-Orr theory, which you've heard much about this past week. And basically, it says that quantum computations within these microtubules are the cause of consciousness. So the collapse of these quantum computations is consciousness. Now, anesthetics would then, which are drugs which erase consciousness, would serve to inhibit these quantum states. Now, the major argument against this idea has always been, isn't biology too warm and wet for these quantum effects? Wouldn't the random motion of the environment wash away all these quantum events so it doesn't render these, makes them ineffective? Now, recently, in the past 5 years or so, there's been much news in scientific literature about quantum coherent energy within photosynthetic systems. Okay? Well, if it can happen in photosynthesis, why can't it happen in microtubules? Okay? Can these support the similar phenomena? And if it can support it, can anesthetics inhibit this phenomena? Now, in photosynthesis, the main thing that causes coherent energy transfer in photosynthesis is a network of chromophoric molecules. These are known as chlorophyll molecules. Everyone has heard of chlorophyll. It absorbs sunlight, and that sunlight gets funneled through protein structures down to a reaction center to give rise to cellular energy and other byproducts. Now, in tubulin, there's a nice network of aromatic amino acids. And here I'm only showing tryptophan. Tryptophan itself is a chromophoric molecule. It can absorb light and it can emit light. Okay? So the arrangement of these tryptophan molecules within the tubulin dimer is very similar to that within these photosynthetic units. The spacing is comparable. The size is comparable. The amount of energy that's required to activate a chlorophyll molecule is about the same that not the energy, but the result, the dipole transition that happens in a chlorophyll molecule is about the same that happens in a tryptophan molecule. Now, what happens is a quanta of light, a single photon comes in, it interacts with the tryptophan's indole ring, these ring structures, and it causes it to jump to an excited state. Now, when it jumps to an excited state, there's a bit of a reorganization of the electrons within the the tryptophan molecule. And this gives rise to what is known as a transition dipole moment. And the transition dipole moment of the tryptophan molecule is about between 5.5 to 6 dpi, which is comparable to that for bacterial chlorophyll or chlorophyll within plants. Now, tryptophan itself is excited within the UV range, so ultraviolet range, somewhere between 260 and 305 nanometers, so a fairly energetic photon. And we can model this, we can form a Hamiltonian of this, so the energy required to excite a single tryptophan molecule, as well as the coupling between these transition dipole moments between tryptophan molecules. And this is exactly the same methodology that they use in these photosynthetic systems. Now, if we take the Hamiltonian that's formed from looking at these properties and move it over into exciton space, so look at the excitations that can form across these tryptophans, we get something like this. So I'm just going to explain this. So here's just a tryptophan within tubulin. This matrix here, each one of these rows corresponds to a single excitation within the within the tubulin molecule. Okay, so this would be one excitation across all the tryptophans. Excuse me. This would be another excitation, but this would be an excitation at slightly lower energy, and then this would be another one at slightly lower energy, and so forth. Now, if you look at the columns, this is talking about an individual tryptophan molecule. So this would be the first tryptophan. This would be the second tryptophan. This one would be the third, the fourth, the fifth, the sixth, the seventh, the eighth, as you go along. Now, the color code tells how much, basically how much excitation each one of these tryptophans have. So if you see white, it's less than 10% excited. If you see red, it's somewhere between 90 and 100% excited. Now, if there's only a single one that's red, then that's 100% localization of the excitation on one of the tryptophan molecules. When you get this blurring, what you're seeing is a spread out of the excitation over several tryptophan molecules. So you're getting an effective delocalization of the excitation. And this delocalization is what is required for some sort of quantum computation. You need to have this spread of the excitation across several of the molecules. Now, in this form, I've shown this before at another TSC conference. This one here takes into account the quantum mechanical shift due to the arrangement of the tryptophan molecules. It takes into account the site energy of each individual tryptophan molecule. As well, I've also taken into account the protein environment. So the protein itself is going to affect how these molecules are excited, and I've accounted for that in this case. Now, so the orientation of the protein is going to have an effect on these site energies. Now, I was talking before about these C-terminal tails. These C-terminal tails stick out from the surface of a tubulin molecule, and they're fairly free to move around. So they jiggle as they function within the cell. So this is, I formed several different formations of the terminal tail. So just taking random orientations of the tail, and for the alpha monomer, and several orientations for the beta monomer, and sandwiching them together, there's about 400 different orientations of these tails. And I wanted to see how these tails would influence these delocalizations within the tubulin molecule. So if we look at, here's the same matrix that I was showing before. Here, like I said, this would be a single excitation on one of the tryptophan molecules, whereas this is a small delocalization across 3 tryptophan molecules. Now, if we look at how the C-terminal tail orientation affects this, we can see that the delocalizations can vary between a single excitation on one molecule or spread out across several of the tubulin the tryptophan molecules. So effectively, these C-terminal tails now can influence the delocalization across the tryptophan molecules within tubulin. This effectively links protein structure to the quantum states within the protein. Now, quantum computers use entanglement and coherence. These delocalized tryptophan excitations could serve as a basis for the sort of quantum computation. Now, this has already been done in photosynthetic systems. They've applied Grover's search algorithms, one of the few known quantum algorithms. They've applied it to photosynthesis to describe how photosynthesis looks for the best path for the photon energy to follow. It looks for the most efficient path, and then it takes that route. A similar mechanism could occur here. Now, you may be still thinking at this point, okay, that's all very nice. You see it in photosynthesis. This can couple to protein conformations. But wait a minute, didn't you say that tryptophan is excited at UV intensity? Right, so is this not, are there UV photons within a cell? Is there a source for UV energy? Because if there's not, then this is all moot. Now, one thing that we do know is, like I said, tryptophan requires UV radiation to be excited. Mitochondria are a source of biophotons around this wavelength. So there's several experiments that have measured UV photons coming from a mitochondria. It's known that some of the processes within mitochondria which produce radical oxygen species, to remove those radical oxygen species, the process that's used to remove those will release a UV photon. So it's plausible that you could have these UV photons coming from mitochondria as a source. So mitochondria could, at one level, produce ATP for cellular energy. At another level, it could be producing UV radiation for use via microtubules. It's also known, Jack's done some work, that microtubules co-localize with mitochondria. So whenever you look within a cell, wherever you find microtubules, you'll find mitochondria very close to these. Now, we were discussing the other day where mitochondria within neurons. Neurons use a high amount of energy, and they rely on this oxidative phosphorylation mechanism from mitochondria. And so the neuron is literally plastered with mitochondria. And I looked this up because I know that we had a discussion, Tony and I. These are found in the cell body, along the length of the axon, within the presynaptic terminal, as well as in dendrites, so in dendritic shafts, but not within the dendritic spine. Right? So effectively now we can link, mitochondria can provide a UV source to power this sort of this effect within microtubules. These excitations can be influenced by the C-terminal tail positions, which would also be affected by post-translational modifications. So you can change the amino acids on the length of these proteins and, again, now affect the tryptophan excitations. As well, the attachment of maps along the surface of the microtubule can also influence these delocalized states. So we can have a way for the cell to interact with these delocalized states, but can these delocalized states then propagate back up to the cell? And the trip dipole, when there's a collapse, the trip dipole is fairly strong. The tryptophan dipole is fairly strong. So it could influence, again, it could be read back out, and it could influence where the C-terminal tail position is going to be. C-terminal tail positions are responsible for many things, including microtubule polymerization, the attachment of microtubule-associated proteins, as well, I was alluding to earlier, the ionic flow around microtubules. So the very strong dipole moment of tryptophan could influence this flow of ions around microtubules, and it could cause perturbations in it, which could then propagate down the length of microtubules and be read out by ion channels or other structures. So here we have computation in tryptophan could couple to microtubule map computation. So we have, at one level, we could have quantum computations, which then would couple to classical computations, like I was showing before, via microtubules and microtubule-associated proteins. What about anesthetics? Okay, so now we have this mechanism. Can anesthetics affect this? Now, one of the things we didn't know for a long time is how anesthetics interact with microtubules. We know that anesthetics do interact with microtubules. They reduce polymerization of microtubules. Halothane can cause an aberrant formation of microtubules. Specifically, halothane is known to bind to tubulin, so they know exactly that it does bind. They just don't know where. And one of the important results that I like is some work by Eckenhoff down in Penn State. He looked at 500 detectable proteins within the brain. And he looked to see how these are affected by two anesthetics, isoflurane and halothane. And he found of the 2%, tubulin is among the 2% of the 500 that is affected by halothane, the 1% that is affected by isoflurane, and it's one of 3 that are affected by both. Okay, so of 500 proteins, only 3 are affected by both, and tubulin is one of them. And tubulin is a very, very prevalent protein within the brain. It makes up the vast majority of the protein within the neuron. But the location and how this interacts is unknown. So we built structures of tubulin and ran simulations of tubulin. So molecular dynamics simulations, all they are is looking at each individual atom in the tubulin protein and applying Newton's equations of motion to this to see how they move throughout time. And what you get is kind of a jiggling effect, and you see the protein conformations and how they change throughout time. We then searched these conformations or these different shapes of the tubulin protein for pockets where anesthetics would prefer to bind. And we looked to see how deep these pockets were by filling them with spheres and measuring the size of the pocket. As well, we looked at to see how hydrophobic the pockets are. So anesthetics, they like to bind in an oil-like region, in a very hydrophobic region. So we looked at how hydrophobic these pockets are, how deep they are, how far away they are from water. And we predicted several sites on the tubulin dimer. In total, we found 47 distinct sites, okay, across all different shapes of the tubulin protein. Only 9 of these sites were found to persist for more than 70% of a 5-nanosecond simulation. So only 9 are persistent sites. Of these 9, key sites are in the GTP binding region. However, in this simulation, we didn't look, GTP was not in the pocket at the time. So when GTP is there, most likely anesthetics aren't going to get in there because GTP is very strongly bound. The colchicine binding site, which is a known MT depolymerizing agent, was one that was predicted, and this is consistent with experimental measures that show that halothane interacts with colchicine binding, as well as the vinca alkaloid site, which is another microtubule depolymerizing agent. As well, several of the zinc binding sites that we had talked about before were also predicted. So another way we can think about this is anesthetics may also have a similar effect on microtubule structures that zinc does. At no concentration, they do nothing. At higher concentrations, they can cause microtubules to depolymerize. If we look at where these anesthetics do bind in comparison to tryptophan, we find that the anesthetics bind as close as 7 angstroms to a tryptophan residue, so fairly close. Okay? Now, if we look at anesthetics, what in the anesthetic? The anesthetics are fairly inert. What could cause these to interact with these tryptophan molecules? Now, anesthetics themselves are known to possess a large dipole moment. So this very large dipole moment could interact with the tryptophan dipole, the transition dipole in tryptophan, either quenching it in certain cases, and that's our belief, or it could alter it in some other way. So these anesthetic dipoles could influence the tryptophan transition and maybe effectively wash out this delocalization. This is something that we're still looking at, that we haven't fully got here yet. So it is plausible that anesthetics could interfere with this tryptophan energy transfer or these tryptophan computations. So in summary, memory depends on the neural cytoskeleton. Using this as a basis that memory is stored within the neural cytoskeleton, we can provide a molecular mechanism of synaptic plasticity and memory encoding, which is so far to this point not fully been explored or hasn't fully been elucidated. It provides a link between the hallmarks of Alzheimer's disease, which no other theory has yet provided. As well, it provides a mechanism for the description of the amnesic effects of anesthetics. So anesthetics affect consciousness, as well they can erase memory, and this is a common site where this could occur. As well, quantum phenomena in microtubules is biologically plausible. Okay, so it is not implausible. It's biologically plausible and could serve as a basis for consciousness, and anesthetics can potentially inhibit this phenomenon in line with the OR-core theory. Most of this work has been published before. Unfortunately, it's cut off here. If you are looking for the citations, you can email me. The stuff on the delocalization across the tryptophan molecules is something that's in progress and should be submitted sometime soon. So I'd like to thank you for listening to my talk and ask if you have any questions.
Yeah, my question is that on the one hand, we look at the activity of a neuron in terms of a binary operation, right? A neuron is either on or off. Mm-hmm. But if within the neuron itself, the amount of information can be, 10 to the power whatever, then how do you square these two perspectives?
Okay, so on the level, so you got to remember that these processes that are taking place within the cell are going to occur on a much faster level than the processes that are occurring at the synapse. Okay, so one that they process much faster to begin with. Now, these structures in the dendrites, they're going to affect how information is received from other neurons. So when a signal is coming in, the changes within these microtubules, which are occurring very fast, can reset up the synapse so that when it receives a signal, now the strength of that signal can either be decreased or increased.
Neuron in the microtubules to process information is tremendous.
Yes.
So there's a kind of a mismatch.
Well, I mean, we would have to look at this building up. So on the level of the quantum level, you have this occurring very fast. This can then affect a slightly higher level, say ionic flow or the tail orientation. This would then affect the way that proteins bind to the surface, which then can propagate up at slightly higher time resolutions. Okay? And as it goes up, it goes up, it goes up, and then eventually you're going to get to the synaptic level.